Good morning, students. Myself, Dr. Ragasudha, Head and Assistant Professor, Department of Microbiology, Government City College, Hyderabad, is here to talk on bacterial toxins. Bacteria have adopted two distinct modes to cause disease in their hosts. These modes are known as infections and intoxications. What is infection? Infection results partially from the growth and reproduction or invasiveness of the bacterial pathogen that often causes alterations in the tissue. Whereas intoxication is the disease which is due to the entrance of a performed toxin into the body of the host cells. Toxin. The term toxin is derived from the Latin word called toxicum, which means poison. It is defined as a substance, such as a metabolic product of the pathogen, that affects the normal metabolism of host cells to such an extent that a deleterious effect manifests on the host. Bacterial toxins can be divided into two main categories, namely exotoxins and endotoxins. Exotoxins are toxic proteins excreted by bacteria into their surrounding media. These are the toxins produced by a live bacterium. Whereas endotoxins are potentially toxic lipopolysaccharides located in the outer membrane of many gram-negative bacteria and released only when the cells disintegrate, or after the death of the bacterial cell, endotoxins will be released into the medium. Let us know the characteristics of exotoxins. Exotoxins are toxic proteins excreted by bacteria into the surrounding medium. These are soluble in nature and heat labile and may travel from the limited site of infection to distinctly located other body tissues or target cells in which they exert their effect. These are inactivated at 60 to 80 degrees centigrade and mostly lethal in very small quantity. These toxins are synthesized by the genes present in plasmids or prophages. Exotoxins are very specific in mechanism of action. Exotoxins are highly immunogenic and stimulate the production of neutralizing antibodies called antitoxins in the host cell. These are inactivated by formaldehyde or iodine and other chemicals, resulting in immunogenic toxides. Good examples for exotoxins are tetanus toxin, diphtheria toxin, and botulinum toxin. Well, these exotoxins are in turn of four types, namely AB exotoxins affecting general tissue, example diphtheria toxin. The second type is AB exotoxin affecting specific host sites, example tetanus toxin and toxin. There are various botulinum toxin, enterotoxin. All these toxins come under AB exotoxins affecting specific host sites. Third type is membrane disrupting toxins, example leukocidins, hemolysins, etc. Leukocidins, they kill phagocytic leukocytes and most of them are produced by pneumococci, streptococci and staphylococci bacteria, while hemolysins disrupt the plasma membrane of red blood cells and some of them also work on cells other than erythrocytes. The fourth type is super antigen exotoxin. These are the antigens that provoke drastic immune response. Certain exotoxins function as super antigens and hence are also known as super antigen exotoxins. Example Staphylococcus enterotoxin, which causes staphylococcal food poisoning. Moving on to the second type of bacterial toxin known as endotoxins. Endotoxins are stated in the beginning, and these are the potentially toxic lipopolysaccharides located in the outer membrane of many gram-negative bacteria and released only when the bacterial cells are disintegrated. Some endotoxins are also released during bacterial multiplication. Bacterial endotoxins are heat stable and pyrogenic, often induce fever in hosts releasing interleukin 1. These are weakly toxic and rarely fatal. They do not convert into toxides. These are these endotoxins are relatively weakly immunogenic. Immune response is not sufficient to neutralize its toxins. These are synthesized directly by the chromosomal genes, unless in exotoxins they are produced by the plasmids. Endotoxins are generally capable of producing systematic effects such as fever, shock, blood coagulation, diarrhea, weakness, inflammation, intestinal hemorrhage, and fibrinolysis. Example for endotoxins are Escherichia, Shigella, and Salmonella bacteria. Difference between exotoxins and endotoxins. In the picture, we can see exotoxins. These are proteinaceous in nature, produced inside pathogenic bacteria, most commonly gram-positive bacteria, as part of their growth and metabolism. The exotoxins are then secreted or released into the surrounding medium following lysis, while endotoxins are the lipid portions of polysaccharides that are part of the outer membrane of the cell wall of gram-negative bacteria. The endotoxins are liberated when the bacteria die and the cell wall breaks apart. Difference between endotoxins and exotoxins in detail. Toxin forms. Endotoxins form a part of cell wall and it is released on the death of the bacteria. Exotoxins are secreted as a part of metabolism. Endotoxins are composed of lipoglycan complex, exotoxins by polypeptides. Endotoxins has an ability to trigger an immune response and is comparatively weak. Exotoxins have a stronger ability to trigger an immune response. Endotoxins, it does not have any enzymatic activity, whereas exotoxins, most of the activities are enzymatic in nature. Endotoxins are moderate toxicity, exo are highly toxic. Endo cannot convert into toxide, whereas exo can convert into toxides. Endotoxins do not prove fatal, whereas exo are fatal in large quantities. Endotoxins does not get denatured, where exotoxins gets denatured on boiling. Endotoxins cause urinary tract infections, coronary artery disease, meningococcal meningitis, whereas exo causes scarlet fever, botulism, scalded skin syndrome. Examples for endotoxin producing bacteria: Salmonella typhi, which causes typhoid; Vibrio cholerae causes cholera; Streptococcus pneumoniae causes sepsis. Examples for exotoxin producing bacteria are Clostridium botulinum, which produces botox; Clostridium tetani causes tetanus; Corynebacterium diphtheriae causing diphtheria. Thank you.